Okay, we discussed up to the vascular events of inflammation. So let me recap very quickly before jumping into the another topic. This was our capillary network. This was arteriole. This was venule side, and we initiated with the tissue injury. Okay, this is the tissue cells. These are the cells. Okay, and we gave an example of injury in our hand. In our hand, there was bacterial invasion going on in this area. So for an example, the bacteria first invaded this particular cell. Okay, this particular cell was injured. Now, as we already know, we have already discussed the causes of cell injury, don't we? Yes, we have already discussed. We also know the causes of cell injury. So one of the causes of cell injury is bacteria as well. The infectious cause of cell injury is bacteria. So bacteria invaded this cell, and it also the all other bacteria, they also invaded many cells. Okay, and these cells were irreversibly injured, and these cell underwent necrosis. These cell underwent necrosis, and their cytoplasmic content were leaked out. Their cytoplasmic content were leaked out, and there were all the features of necrosis. We have already discussed the morphological features of necrosis, those ground glass appearance, and all those things, eosinophilia, everything. So this cell now, they underwent necrosis, and they will now elicit inflammation. They will now elicit, or they will now bring inflammation. Now how do they bring inflammation? Same thing we are studying. We are studying the process of inflammation. How does the inflammation happen? These all things we are studying. Okay, these all things are interrelated. So now these cells are irreversibly injured, and they underwent necrosis. And now what do they do is, there is another type of cell also in this tissue. These are mast cell. Now mast cell, they are always present in the mucous membrane, in and around the blood vessels, and below the skin. Now where are the mast cell present? Mast cell are always present in and around blood vessels, below the skin, and in the mucous membrane. Now this mast cell, they secreted histamine, and the injured cell secreted PG, prostaglandins, and the endothelial cell, they secreted NO. And all these three chemical mediators of inflammation, they created arteriole dilation. They created increase in the hydrostatic pressure. They created exudation, and also, okay, it can be those bacteria also damaged this endothelial cell, and they created what? Capillary permeability. And maybe those histamines and whatever causes we discussed in the last class, those causes, they created capillary permeability, and they also created exudation. Now what up to here we were clear. What is exudation? Now again, exudation is the escape of protein rich fluid. After some classes we'll discuss the difference between exudation and transudation, but for now, in inflammation there is exudation. This thing you need to be clear. In inflammation, in the process of inflammation we're talking about, there is exudation. Up to here it was very clear. Now I'll continue from exudation. Okay, now let me start with this one. See, it was already what? Arteriodilation was there, exudation was there. Now what happens after exudation is, exudation means what? There is protein rich fluid will get outside. If there is escape of protein and plasma from blood to the tissue, then what will happen? The blood will contain more formed elements. Blood contains two things: plasma and formed elements. Now plasma has escaped. Formed elements, those RBC, WBCs, they are very huge. They are large in size, so they cannot escape through intraendothelial gaps. So they remained inside. inside. RBCs and WBC remained inside. Now whatever events that are happening, we'll talk in cellular events. Now RBCs and WBCs are already inside this capillary membrane, inside this capillary. So this, now what happens is they gets concentrated, so that thing we call is as hemoconcentration. So due to concentration of formed elements, blood becomes viscous and the blood will now slow down. So there will be RBC clumping now. There will be RBC clumping going on. And due to clumping of the RBCs in the center there is complete clumping of the RBCs get clumped at the center. So this will push the WBC to the periphery. And this process is known as margination of WBC. This process is called margination of WBC. Now what happens after margination of WBC develop a special kind of selectins, special kind of sialylated sugars they develop in their membrane in order to get attached to the endothelium. Now our target is to bring out WBC from this blood to tissue, where their fight is going on, between the bacteria, and now we're talking about tissues. Yet there is no any WBC there. Maybe some tissue histiocytes, macrophages are working there. They are just fighting to the bacteria, and they are calling for backup. Now our backup is our WBC that is running in the blood. So now that WBC needs to get outside the blood and reach to the tissue. Now WBC up to now has been marginalized. They have been marginalized up to now. Now they develop a special kind of selectins in their surface. See, I have mentioned this is what? Neutrophil. Because in neutrophils are the first WBC to reach into inflammation. This is also basic thing. Neutrophils are the first WBCs that come to the inflammation. They are the first. They are the first policeman. They are the first policeman that come to the inflammation. So WBCs came, and especially neutrophil came, and they developed L-selectins. Okay, they developed L-selectins, and the endothelium also developed P-selectin due to histamine, leukotrienes, and thromboxans. Histamines are released by mast cell. Okay, and these developed P-selectins and L-selectins, and now this WBC they roll upon the endothelium, because this selectin interaction is very, very poor interaction, very, very weak interaction. Selectin interaction is very, very poor interaction. That's why WBC and this endothelium cannot stick, but they will roll. They will roll upon the WBCs. They will roll upon the WBC. Now again, rolling is going on. But let me remind you another thing. Histamine, leukotriene, and thromboxane, they develop P-selectins, and after some time, interleukin-1 and tumor necrosis factor, they develop E-selectin. But may it be E or P-selectins, they are not forming any strong interaction to the WBCs. So these WBCs and endothelium, if they have selectins, then they are not strongly connected. They are not strongly holding each other. That's why they roll. Now after some time, they develop another type of special proteins. They're called integrins. Now they develop another kind of special proteins. They are called integrins. Now they WBC develop integrins, and now these endotheliums they develop ICAM and VCAM. They are also integrins, and their name is vascular cell adhesion molecule. Itself, their name itself suggests adhesion. It's not rolling, okay? It's not rolling. See, I'll tell you from the beginning. What are the process we discussed? First, we discussed margination. WBCs were marginalized. Second, we discussed rolling. They will roll. Now, thirdly, they will what? They will adhere. They will adhere. Now they will adhere. Now tashinsan. Okay. Now what happens is those adhesion is mediated by integrins of WBC. These are neutrophils, again having three lobe nucleus. Okay, these are neutrophils. And now what they do is integrins and ICAM they will adhere. They will adhere. What is ICAM? ICAM is intercellular cell adhesion molecule. Okay, these are intercellular cell adhesion molecule. Now imagine every WBCs were marginalized. They were going to the margin. They were rolling and rolling on the epithelium, and now they are halt. Now they are integrated. Now they are adhered. This is called pigmentation. This is called pigmentation because many WBCs will now adhere to the endothelial membrane and pigment of WBC on endothelium. They will do pigment of WBC on endothelium. Now what happens? Now they are now paving the endothelium. Now they need to be extravasated. That's called extravasation. That is coming out of WBC from the blood to the tissue. That is known as diapedesis. Diapedesis, also known as transmigration. This thing now we'll discuss. How does transmigration happen? Now they are already adhered. They are already adhered to the to the endothelium. Okay. Now what happens? Okay, now for an example, this is one endothelium. They are contracted. Obviously, we have already discussed the contraction of endothelium. They are contracted. Now there is interendothelial gap. What do? Is they both develop similar kind of these attaching molecules like this. So now what they do is this two endothelium will now catch this one and it will pull down. It will pull this WBC inside the tissue. Okay. Now this is mediated by this production. Now what is first I'll tell the name of these molecules. This molecule is PCAM. It's not ICAM, it's not VCAM, it's PCAM now. Okay. This thing you need to now mug up. Now what to do? Platelets endothelial cell adhesion molecule. Now this will bring this WBC towards the tissue. Okay, now these are mediated by cytokines mediated PCAM development. Let's write this one. Now PCAM causes diapedesis. Now ultimately WBCs has reached towards the now basement membrane. We have basement membrane also here. Okay. Now basement membrane contains collagen and WBC will secrete collagenase. WBC secrete collagenases. This will dissolve basement membrane. Now it reaches towards the subendothelial area now. That means it will now reach towards the tissue. Okay. There is nothing below the endothelium in the capillaries. Now it will reach towards the tissue. Now WBC is finally in the WBC. Okay, specifically we can say neutrophils. Neutrophils now in the tissue. Now they need to find where are the bacteria so as to engulf them. so as to engulf them. So this thing is done by chemotaxis. This thing is done by chemotaxis. Okay, now let's talk about chemotaxis. What are the factors that is present in the bacteria, or what are the factors that are released in inflammation, so as the WBC will rule out the site of inflammation. Inflammation कहाँ भइरहेको छ भन्ने कुरा थाहा पाउँछ. These are known as chemotactic factors. These are called chemotactic factors. What are they? Their first one is bacterial products like N-formyl methionine. Many bacteria has this product. This is the bacterial product. Second thing is leukotriene. Leukotriene B4 also act as chemotaxis and complement product. I hope complement system is covered in microbiology. Complement system product C5A. I'll be also covering complement system in the chemical mediators of inflammation. Okay, in complement system, C5A also acts as chemotactic agents and also interleukin 8. Now what do? Is they attract the WBC towards the site of inflammation. Okay, now these are the things we are discussing up to here. Now after this, now they are coming in site of inflammation. Okay, this is the site of inflammation. Okay, and now WBC are there. Okay, neutrophils are there now. Now how do this neutrophil recognize the bacteria? Okay, these are the bacteria. These are very bad, and these are very badass bacteria. Now this bacteria, how do this neutrophil recognize these offending agents? This we'll discuss in the next video.